Good afternoon, everyone. I'm going to be talking you through this small project that I worked on in collaboration with Steve Summers at Celsi, looking at rapid nanopore cloud-based monitoring and analysis of commercial aquaculture microbiomes. Ordinarily, I do work at SMART, and I do more sterility-focused work, but this is a small talk that I wanted to share with you. Okay, and this would not have been possible without the funding from the Singapore National Biofilm Consortium, as well as the academic team and the industrial team from the Singapore Aquaculture Technologies, which we'll see more of in a moment. So why monitor aquaculture facilities? So in the case of this work, the industrial partner that we were working with grow barramundi or Asian sea bass on a barge, and that's what you can see here in the image. And they want to do so in a sustainable way, so they do use a closed culture system as opposed to an open water culture system, which means that they grow their fish in tanks, which are climate controlled essentially. And so this is all part of work for the Singapore 30 by 30, which is to produce 30% of Singapore's produce by 2030. And so because this is a living system, it's important to understand how that will change over time. So the main goal of this work was to lay down a metagenomic baseline to understand how this aquaculture system works in a healthy setup. And so as part of this, we also want to understand going forwards as well, in terms of biosurveillance, what are the threats that the fish can have, and what organisms can grow that can be pathogenic. And so in order to study this, we then look to the microbiome. And so the microbiome, you can examine it in a number of ways in this particular context, which is to look at the practices, the fish treatment, infection events, as well as the water quality. And to highlight a few here, so one of them can be this fish transfer, where you start with which is simply the difference between where you rear the fish and then where you grow the fish later on. Another example would be like the rearing density, and then the feeding in terms of what diet are you giving them, how are you modifying it, are the fish starving because they haven't got enough. Also the treatment of the fish, so this is primarily antibiotics to treat any infections that you will get at these very high densities. And then finally, the water quality, which includes things like dissolved oxygen, pH, nitrates, and salinity. And then, of course, the actual potential contaminants, which could be bacteria, fungus, viral, or parasites. Okay. And before I go forward with the actual data, I'd just like to give you some perspective on the current economic aquaculture landscape in terms of disease. So 2 years ago, there was this paper published on scale drop disease virus, specifically in barramundi. And then for Singapore, especially more recently, there was this recent article here about the barramundi group who are having a massive outbreak. But as you notice, they are growing it in open water setting as opposed to closed water setting. And in fact, a month later, they then declared that they would be moving their operations to Brunei because it was no longer tenable in Singapore. And so, as part of the learning process, Singapore is also reaching out to other countries that have their own aquaculture sectors, such as Australia, to learn from them and to improve. Okay, so this is the aquaculture facility with the industrial partner that we worked with. It's a large barge, about 30 meters by 100 meters long. It's comprised of these 10 of these blue tanks, which can each hold 20,000 barramundi fish, and they're about 5 meters deep by 5 meters in diameter. And you can see down here, this is on the right and the left where the young fish will be reared, and then you have various steps of biofiltration, which ultimately ends up in this tank. And so this is the sampling workflow that we went with. You can see here the diagram of the barge with the 3 sampling locations. So what this means is that we have the drum tank where we collect scum from the side of the tank itself, which will be like a biofilm that builds up, as well as collecting samples from the sump, which is the lowest point in the system. It's where the detritus will all filter down and be collected. And then finally, the biodiscs, which are these high surface area objects that float within the water itself and help to maintain the nitrogen cycle. And in terms of the sample preparation, it was a relatively simple workflow because the primary goal here is that the company would be able to run this on the barge on site and do it as quickly as possible. And so to that end, we have a relatively simple DNA extraction using mechanical lysis, followed by the rapid sequencing kit to sequence your reads and then uploading that data to a server for data analysis. And just to hammer home the point, this is relatively rapid. It can be done between 7 and 10 hours, depending on how long you want to sequence your samples for. And in terms of the bioinformatics workflow, it's also relatively simple as well to get a rapid result in terms of you start from your Nanopore electrical impedance signal, you complete the base calling, you clean up your reads, and then you remove the host reads, which in this case would be the barramundi reads, before moving on to metagenomic classification using a viral, fungal, and bacterial database to identify the metagenome that is present. And then we also perform some additional statistical analyses looking at the diversity and the coverage. And so just before we jump in a bit more, I want to give you a bit of background in case you're not familiar with some of these terms. So alpha and beta diversity, in this context, the alpha diversity is the variance within a single sample, while the beta diversity is the pairwise diversity between two samples. And so to this point, from the sampling locations that we examined with the samples that we had, the take-home for the alpha diversity, which is the intra-sample diversity, is that they have relatively a similar composition within the samples. But more interestingly, when you look at the sampling locations themselves, they have much more diverse species composition, which you can see on the right side here with the beta diversity plot. Okay, and so for the remainder of this talk, we're going to focus entirely on nitrogen-associated organisms, just to drill down into a subset of the predicted species. And so that's what you've got here. The take-home from this heat map is that the biodiscs contain the highest frequency of the reads, and that the nitrogen-associated organisms are identified the most within these biodiscs, which you can see here, especially in the later samples that we collected, where we have the highest number of reads associated with these species. You can also see that the drum tank appears to be the most poor in terms of having these species present, which is interesting because they're relative physically quite close to each other, but they don't provide that same niche that the biodiscs do. Okay, and so now I'm going to drill down into 3 examples here, just to show you what these individual samples look like. But before we do that, I'm also going to tell you a bit about how we're going to look at them. And since this is not amplicon-seq DNA, this is just native DNA sequencing, we don't necessarily have amplified ribosomal RNA sequences, but they are still present within the sequence data, especially here where we can look to target the 16S and 23S, or the small subunit and the large subunit, which are shown here on the diagram. And you can see the 16S is relatively smaller than the 23S. And if you wanted to do the same thing with the eukaryotic sequences, you could do, but the sizes are different. Okay, so we'll be focusing on the top 2 elements. And then before moving on to the data, I just want to also explain the concepts of the region bias and the percent identity. So these are measures of the breadth of your sequencing. So on the top here, we have basically very low breadth of sequencing, maybe 30% aligned with the reference genome, and then here we would have 100%. And then on the right side, we have the sequence similarity, which is where we compare the individual bases between the reference and the reads at the base level. Okay, so now we have a comparison across the 3 exemplars that I showed you earlier, where we can see that the biodiscs have the highest coverage. But before we get there, so first off, we have here the organisms on the left side of the heat map with the relative abundance within that population, within that sample, and then the actual number of reads. And then on the right here, we have the small subunit and the large subunit. So the way to interpret this plot is that if we take this example here of 70%, what this means is that these bases are 70% similar to the reference genome at the individual base level. And then on the left here, this is a ratio out from 0 to 1, where 0 is no similarity and 1 is complete similarity. Complete coverage or breadth. And so what we can conclude from this is, in addition to the read sequencing data from the metagenomic classifier now we have this coverage information, we can be slightly more confident in saying that, that these 3 organisms at least are present, and then similarly in the drum tank. But the interesting part is when we now look at the biodiscs, we can see that there are more organisms and that a lot of them do have complete coverage across the reference genome and also are quite similar with the reference genomes that we are using. Okay, and so in addition to this, we also did a network analysis of taxa co-occurrence in the aquaculture microbiome. And the key finding here is that within the other 2 sampling locations of the sump and the drum tank, there was relatively little networking involved. However, in the biodiscs, what we observed are these, and there were 8 samples present within the biodiscs, these 2 populations of organisms, which suggests that there is some kind of niche within the biodiscs that may be forming, either perhaps due to anaerobic conditions or aerobic conditions, or as seems to be the case based on these organisms, it's probably something a bit more to do with denitrification versus nitrogen fixation. And so moving forward, this was only a 6-month project, and we want to move it to a more complete piece of work. And so for that, we've applied for additional funding for this AquaBoost project, where we'll be trying to expand our knowledge and increase the understanding we have of these aquaculture microbiomes with the goal of generating a product that is relatively simple to use, rapid and real time. And of course, this also could include understanding viruses, but as some of you may be aware, it's much more difficult to work with viruses than with bacteria and fungus, at least for metagenomics. And with that, I'm done.
Okay, are there any questions from the floor? Okay, I'll start that actually. So actually, I'm curious, because you showed a plot where essentially it's around 10 hours for the whole process right now, is it? So does sort of the data analysis have to wait for the sequencing to be done, or is it done like real time, for example?
So we haven't implemented it to be done in real time, but that definitely already exists for Nanopore products. So it could be accelerated perhaps by another hour by doing that. Okay,
yeah, and I think that's actually quite crucial, because if I understand correctly, like some of the outbreaks of diseases and things are quite time sensitive.
Yes.
So actually, at the end of the day, every hour that you save you're saving quite a lot of money as well.
Yeah, of course, part of this question is how willing is the company to test every day versus once a week, because you would probably have to do that if you wanted to capture something early. Otherwise, you're relying. So I should have said as well that the company also does chemical and physical environmental testing.
Oh, okay.